So DNA polymerase, okay, the enzyme that's going to link together the nucleotides, does so in such a way that it utilizes a template strand to add the complement strand. Remember here, the complement means that if the template strand says T, the complement will be A. Template strand says C, complement will be G. And it's able to do so, look at that, 500 nucleotides per second. I'm sorry, I was off. I have been saying hundreds of nucleotides per minute. I was wrong. It's hundreds of nucleotides per second in bacteria. For us eukaryotes, it's actually about tenfold lower at about 50 per second. Why so much slower for us compared to bacteria if we're supposed to be the more advanced thing? Well, because bacteria divide, remember, binary fission, they replicate once every 30 to 60 minutes. So if they screw up and it's a mutation that's bad, well, not a problem. 30 to 60 minutes from now, they'll make another. And if it happens to be good, well, this mutation could give that bacteria a new ability or a new resistance or something like that. It's a single cell organism, so if it screws up one, no big deal. There'll be another in half an hour. Whereas for us, we are multicellular organisms. We cannot risk an alteration of the chromosomes. An alteration of the chromosome sequence, the DNA sequence, is a mutation. Okay? Mutations could be bad. Think back. A mutation in a certain gene could lead to the loss of checkpoint control, which means cancer. Cancer will kill the organism. So we want to maintain accuracy, maintain fidelity. So our polymerases go slower. Plus, mitosis in us can take 1 to 2 days. So it's not something that's going to be happen every 30 minutes like for Staphylococcus aureus. Notice orientation. Okay, the way these sugars are pointing. Okay, 3 is at the bottom, the 5' that heads the phosphate is up. So this strand's pointing this way, this strand is pointing that way. Antiparallel. Where the template says T, new strand will lay down an A. Where it says G, the complement will be C. This open 3' end right there is necessary. DNA polymerase can only add a new nucleotide to an open 3' carbon on a sugar. Okay? That's why you have to have the RNA primer. Primase can lay down a primer anywhere. DNA polymerase has to have that open 3' to build on. DNA polymerase allows any nucleotide to enter in, but only the nucleotide that can hydrogen bond to the template will remain there and then be acted upon. What's going to happen? Cut right there. The second and third phosphate will be removed, and that phosphate that we see right here now becomes this phosphate right there. The second and third phosphate off of that nucleoside, remember, add a phosphate to a molecule, to an enzyme, you imbue energy. When you transfer out that phosphate, you transfer energy somewhere else. Well, the second, third phosphate energy is what is used to add this nucleotide to the growing new strand. Now, what's interesting is you have this addition of new nucleotides happening in 4 places for every replication bubble. You have one here, down here, up here, right there. Okay? 4 places that you have all of this machinery with the polymerases at work. But we run into an issue. What we run into the issue is, well, you start right here with this primer, and DNA polymerase can run this way because the leading strand is in the opposite direction from the replication fork. But this template strand is in the wrong orientation. It cannot be read from the origin out. No, it has to be read from the replication fork back towards the origin. And the opposite is true. You have on the other replication fork, one side that can be read, the other the wrong orientation. The template strand that can be read from the origin towards the replication fork, heading out, that is known as the leading strand. This is the simplistic, easiest. Primase lays down a primer, and then DNA polymerase starts and just keeps going. Okay? The lagging strand, as we're going to see, well, here there's going to have to be some more steps taken involved because everything is backwards. So with the leading strand, literally, like I said, RNA primer lays down the short primer that's needed to give a open 3'. DNA polymerase 3. Shorthand DNA Pol 3. Pol means polymerase. And DNA polymerase is held to the strand by a clamp protein. Holds it in place, keeps it there, keeps it going. Helicase unzips, and then along comes polymerase. Easy peasy. Okay, leading strand here, leading strand there on opposite sides, catty corner. Easy as pie. But as we're going to see in the next lecture, the strands where it's in the wrong orientation, thus polymerase can't just start in one place and run through. Well, these are going to be called lagging strands. And if you go back to the Canvas webpage for this chapter, you will see that there are some YouTube videos that are really good at illustrating this process. The lagging strand gets its name because it lags behind. It's not as fast as the leading strand. Japanese researcher by the name of Okazaki spent his research time studying how this DNA at the replication fork that's in the wrong orientation, how it is able to, be replicated, how the template can be read and the new strand made if it's backwards. He was able to show that the leading strand, that when he isolated the DNA from the leading strand and ran it on a gel, that given time, that new strand just kept increasing in size. It correlated time to size. Everything was good. But when he isolated DNA from the other side, that the strand that's in the wrong orientation, he noticed that yes, there was a part that kept getting larger and larger, but there were always these small little fragments. Small fragments that seem to be maintained, the numbers maintained, the sizes maintained. That even though the replication fork would continue moving and that continuous new template would continue to get larger, there would always be these fragments, which we now refer to as Okazaki fragments.